Hello again. This video on chloroplasts is going to focus on the structure of chloroplasts. I will drop some hints towards the end of the video on photosynthesis and how the structures relate to the function of photosynthesis, but the topic of photosynthesis is a big one and it's an entirely different video. On the board right now I've represented both cellular respiration and photosynthesis as a cyclic process. If we look at this one here, this part, this is what we would consider as respiration. Where we start with glucose and oxygen and we wind up with carbon dioxide and water. And what we also recognize is that the product of ATP, adenosine triphosphate, is generated along that process. That's respiration. That's not this topic. This topic is chloroplasts, and the function of photosynthesis is represented this way, where we start with carbon dioxide and water. We can cleave off some oxygen and we wind up with sugar. Of course, if we have energy as a product in here, we have to have energy as an ingredient or as a reactant in here, and that's where sunlight fits in. So this is photosynthesis. Now, mitochondria look after this process here. Anaerobic respiration occurs in the cytoplasm, but most of the ATP is generated inside the mitochondria, the powerhouses of cells, and chloroplasts look after this process. So you might suggest a similarity between the two, and there is a similarity between the two. We represent mitochondria this way as elongate organelles where the inner membrane forms these cristae, these shelf-like processes that extend into the matrix like that. Chloroplasts, in contrast to that, are much larger and they're visible. They're visible because they have chlorophyll in them, and that gives them a green coloring. These, as we've said before, are quite invisible. You can't see those. So let's look at the similarities between mitochondria that way and chloroplasts, and let me just make some space for that here. At first glance, they're both organelles which have an outer membrane. I'm going to represent a chloroplast as a larger organelle. It is larger. The outer membrane of the mitochondria and the outer membrane of the chloroplast are similar. They both have, see, I'll put outer membrane. They both have porins. Reminder, porins are proteins. They're the proteins that line the pores, and hence the word porins, and they would allow molecules to enter, even protein, large molecules to enter into the chloroplast. This one also has porins as well, so it's a similarity that exists in both of them. The mitochondria has this very convoluted inner membrane that gives it a lot of surface area because there's a lot of really important chemical reactions occur along that membrane. The chloroplast also has an inner membrane, but this inner membrane is not convoluted. Instead, I'm just going to erase the tip of that arrow because the porins allow the proteins into the intermembranous space, but not suggesting that they cross the inner membrane. So we have an inner membrane here. Not convoluted. It doesn't have that huge amount of surface area there. It doesn't have the porins on it. So right away you recognize that it has a different function. It has a different structure than the outer membrane, hence a different function. It does have permeability. All membranes have a certain degree of permeability, and that's a function of the proteins that are in it, but it doesn't have these protein-lined pores. They're lined with the porins. Mitochondria, we say it's got this huge surface area in its inner membrane. These have a third membrane that has a huge surface area. And I'm going to represent it like this, and then I'll show you the origin of this membrane. These membranes actually form disc-like structures. And I'll put another set of them over here. So if you were to look at a chloroplast, really magnified, you look at it under a microscope, really magnified, you'd see that you have this type of an arrangement here where you have the outer membrane, the inner membrane. This is the fluid here. This is called stroma. Now that would correspond to the matrix here in the mitochondria. But this is stroma, and this additional set of membranes inside here is a bunch of disc-like things. Like if you were to take a series of coins and stack them all up, that's sort of what these things are like. They're little discs like that. Each one of these things is called a thylakoid. And they are hollow. They're just a membranous disc. And I suppose I could take one and just show you quickly, if we were to take in 3 dimensions and cut it open. That's sort of what it might be. So we have the membrane on the outside, and inside there's a space, a fluid-filled space. It's called the lumen. The lumen is a term that's used for it's the space of a hollow structure. Lumen. So that's a thylakoid like that. So we have a thylakoid membrane that's in this sort of thing. If we want to talk about one of these disc things itself, it's called a granum. So we have the thylakoid membrane, which comprises a granum, and if we have a stack of them, plural of granum is grana. So we have a stack of thylakoid membranous structures, each one is a granum, and they're interconnected. So there's another structure in here, another membranous structure. There's a membrane that will actually interconnect these things, so that maybe there's another one over here, another set. These things are called the lamellar membranes. It would be unreasonable to think that these stacks of grana, these granums, or the grana, would be loose in here. They're actually interconnected. They don't rattle around inside this otherwise fluid-filled hollow sort of structure. So then we have a very big distinction here between mitochondria and chloroplasts now. We've got a third membrane, and that doesn't exist here, but it does provide that huge surface area because it's this membrane, the thylakoid membrane, that has all the reactions and processes going on in it. I should mention as well that in the stroma there are ribosomes, there are enzymes in here, there is a genome. Remember, a genome is the DNA, and that's not as well drawn as it could be, but it's circular DNA. Same as the genome inside mitochondria. DNA we would represent typically like this alpha helix that does that. Well, it's a circular DNA structure in here. And there's multiple copies of it. When you recognize multiple copies, you recognize that, okay, they can reproduce. They can make more of them. Okay, so right away, structural similarities, outer membrane, inner membrane, lots of surface area here, there's lots of surface area there, but it's provided by the third inner membrane. They both have circular genomes, enzymes, ribosomes, fluid-filled interiors, the matrix, and stroma. So a lot of similarities between them, but at the same time, you see there are some very significant differences. I want to come back to this just one point here, is where does this come from? And I'm just going to steal this space here. And if we think for a second about a branch on a tree, and the branch has got a little tiny leaf bud that's starting to grow into a leaf. And if you leave it long enough, that leaf actually develops into some sort of leaf, larger structure. To get from here to here, cells have to be produced at an incredible rate. And these cells, these plant cells and leaves anyway, have chloroplasts. So obviously the chloroplasts have to be produced at incredible rates as well. Chloroplasts originate from plastids. Now a plastid itself is just this much. It doesn't have all that stuff inside. It's not equipped with it. It can generate it. It's got the enzymes that allow it to generate the chlorophyll and that sort of thing. But the plastids themselves don't have all of that internal membranous structure. What happens, or what seems to happen, is that this inner membrane, I'll just take a little piece of it off here, this inner membrane forms a saccule. And we've seen this kind of a process before where it forms a bleb or a bud, and if that's what it looks like, then the next one, or the previous one might look like that. So it'll form a series of these things. That's how these things are generated. So where the inner membrane here does provide all the surface area, the inner membrane here doesn't directly provide the surface area, but it gives rise to all this thylakoid membrane, the structure, the grana inside. So it does give rise to all of that surface area. Okay? So structurally they are related, but they're not exactly the same. Now I do want to mention a little bit about photosynthesis, although the function of this video, or the purpose of this video, isn't to direct our attention to photosynthesis, but I do want to mention a little bit about it, and I think maybe I can go back to this picture here. What happens in photosynthesis, I'll just get back to where we were here. If we have, well, I can use this here. What happens, we get light shining on a leaf, and we have a chlorophyll molecule. Now you'll say, well, where's the chlorophyll? The chlorophyll is located inside this membrane. So chlorophyll is a component of that membrane. So you can see that the arrangement of the lamellar membrane and these grana, it sort of spreads it out. So they provides enough surface area for the light. It maximizes light potential for light to strike as much of that membrane as possible. Well, what happens when light shines on chlorophyll? So the light, I'll put the light here now. It's shining on the leaf, but when the light shines on chlorophyll, chlorophyll is a very large molecule. What happens is it releases an electron. And without the electron, by releasing the electron, it gives the light energy is transferred to the electron. And then the electron has to be returned back to either the same chlorophyll molecule or a different chlorophyll molecule. In doing so, what happens is it generates this energy here. It generates enough chemical energy, whichever one we're talking about, to make ATP. Now that's a similar process as what went on in mitochondria. Remember, the electron transport chain in mitochondria does the same sort of thing, where it takes the electrons, it moves them through a series of cytochromes, and it makes ATP. Well, the same sort of thing here, except this is the first step here. And what happens in photosynthesis, if this is the first sort of thing that happens, this is called what we call the light reaction, because it's light dependent. It originates because of light. The second thing, if this is the light reaction, the second one is often called the dark reaction because it's not dependent on light. It doesn't necessarily need the absence of light. It can occur when light is present, but it doesn't depend on light. So it'd be a bit more correct to say the light-dependent reaction and the light-independent reaction, because this will take place in light and in dark. But what happens here is that this ATP that was generated is used to take carbon dioxide and convert it into sugar. Now you can see right away this has got carbon and oxygen in it. Carbon and oxygen here, we have to have hydrogen added to it. So with the combination of carbon, hydrogen, oxygen will give you that. These, you know, the atom numbers of atoms aren't balanced here, but you get this sort of thing here. This is called the Calvin cycle. So this topic is a topic of a different video to get into all of the details of how these reactions actually occur. But the reason I want to mention it now is because I'm trying to draw contrasts and comparisons between what goes on with mitochondria and chloroplasts, and the electron transport chain or the electron transport system that makes ATP in chloroplasts occurs first. And it's followed by a cycle known as Calvin cycle, which makes the sugar here. And the contrast here with mitochondria, we had Krebs cycle, which occurred first, and it was followed by the electron transport chain. So, I think that does it for this video. I'll put a link up to the video that dealt with mitochondria, so if you aren't sure about some of the things I said about mitochondria, I'll put a link to that video up here at the end of this one, and you'll be able to see that. Of course, my subscription gecko, I'll put it back down here in the bottom right corner. One other thing I should mention, I could mention, this is a cycle in here. You can see that's cyclic photophosphorylation, or a cyclic process. This one is a non-cyclic, so we have a non-cyclic photophosphorylation. So that topic, like I say, that's a different video in a different video series. Thank you very much.